Topic 15b, mutation in DNA repair. DNA mutation is responsible for the variety we see within and between species. For example, mutations are the basis of different gene alleles that create the different head and body shapes and coat color patterns between different breeds of dogs. A genetic mutation is a change in the nucleotide sequence of DNA. For example, the GC pair in the upper strand has been changed to an AT pair in the lower. Otherwise, the strands are the same. So, how can mutations occur? We are already aware of DNA polymerase's ability to add new nucleotides and synthesize DNA. Sometimes DNA polymerase will make a mistake and pair the wrong nucleotide. Most of the time, the DNA polymerase will fix this with its 3' to 5' exonuclease activity. This means it can back up, remove the nucleotide, and try again. Sometimes, however, these mistakes are missed and the nucleotide change can become permanent. Another source of mutation is unpaired oxygen atoms. These will bind to guanine to make a nucleotide called 8-oxoguanine. 8-oxoguanine pairs with adenine instead of cytosine, which creates a change in the nucleotide sequence. Ultraviolet light also damages DNA, which can lead to mutations. UV light is absorbed by thymines, and if two are next to each other, they will fuse, creating a covalently bound dimer. This dimer changes the shape of the DNA molecules, so DNA replication cannot proceed normally. 8-oxoguanine and UV light damage would be repaired by a process called excision repair. In this process, an enzyme, represented by this arch, cuts the phosphodiester bonds of one of the strands of DNA and removes a group of nucleotides containing the damaged area. DNA polymerase will then come in and fill the gap. Another form of energy that damages DNA and can lead to mutation is ionizing radiation like gamma radiation. This energy will pass through a cell and shear any DNA molecule in its path. This type of damage would be repaired by the enzyme ligase, which would reform the phosphodiester backbone. To review, mutations are the source of biological diversity. A mutation is a change in the nucleotide content of DNA. Mutations may be caused by mistakes during replication, chemical modification of nucleotides, or external energy. Most mutations are repaired, but not all, and they become permanent.